Yeah, that was quick. Thanks. Thank you very much. It's great to be here. It's nice to be here in a professional manner. I've only been here as a tourist before, so it's fun to see the campus and inside the buildings as opposed to just a kind of college tour around the outside of the buildings. And so yeah, so the title's probably not the same because I think it's one of the few things in this talk I forgot to change since I gave this talk uh, last week, as it turns out. But I've added a lot of new data here. So there's a lot that I haven't presented before. So I'm interested to see kind of how it goes, both accepted and how I managed to stumble through it. So it should be hopefully fun for everybody. So let's see, change this. Okay, so some conflicts. Um, so our lab is really focused. It's kind of has two main themes, if you will, in our lab. We're interested in both chronic virus infections and cancer. We also have a bunch of other interests, as it turns out, but they just haven't kind of cracked these top two yet. And we're really interested in these two because kind of how the immune system functions in times of chronic stimulation. I think something it's probably not really built for. I think it's learned to adapt, but probably not kind of wasn't made for this kind of an immune response, certainly against kind of high level, high antigen, high stimulation type of attacks. So we really try to understand kind of both the shared and then divergent mechanisms of how the immune system is working in these type of chronic antigen activation states, with the hope that we could find something that would work across the boards to treat these diseases, or something to treat either one, or at the very least to get a kind of a basic understanding of how the immune system performs. And our lab has very much a T cell focus, although we've been known to have affairs with other cells and certainly have published on a bunch of other cell types and how they're affected in these type of diseases. So I just kind of, when we talk about chronic antigen, chronic stimulation, kind of this idea of immune exhaustion, a lot of times we focus on T cell, CD8 T cell exhaustion. And I think this is fair. This is the end effector. These are the cells that are ultimately going to kill a cancer cell, a virus infected cell, and their loss of function, or in the case of really high affinity CD8 T cells deletion, certainly has an impact on the ability to control these diseases. In fact, this is where, probably not solely working on these, but the thought is this is where a lot of these immune checkpoints that are currently in the clinic, and I think have been very successful in many ways, are thought to act. But the truth is, in these times of chronic diseases, you get really a systemic change in the immune response. You get changes in the innate immune cells. So dendritic cells, macrophages go from being really highly stimulatory towards many of them being immunosuppressive. This can then have effects both on the other cells as well as secondary infections that would take place when you have either a chronic virus infection or cancer. You have changes in CD4 T cells, both in their function as well as the subset they differentiate into. You have changes in B cells that I think are less well characterized. I keep saying that this is something that we're going to learn a lot about soon, and very soon we'll understand this. I think, well, I've been saying that for many years now, and we still don't. So maybe we'll never understand what these are. Hopefully some of you out there are working on this and can kind of clarify what these B cells are doing for good or bad in the chronic virus or tumor microenvironment. And of course, changes in infected cells and tumor cells, so that you get they can become resistant to immune killing through the upregulation of various surface or soluble markers that can both affect the environment and affect all these things, as well as directly prevent these CD8 T cells from killing them. And so you get this systemic change. And what got us really interested kind of in this field of interferons was that interferons underlie a lot of these immune dysfunctions. So they're not the sole culprits, of course, but they are really, in many cases, big contributors to kind of all of these changes that are observed. And so what are interferons? Type 1 interferons. So when I talk about, there's a variety of them. When I talk about them, I mean interferon alpha and interferon beta. These are made in a variety of ways. They're made in response to virus infection is really how they're best known to be induced. Their antiviral qualities are certainly how they were discovered. Virus infection triggers changes or recognition by various pattern recognition receptors that lead to the upregulation of interferons in this case, as well as any kind of change in the cell. So cellular disequilibrium, changes in kind of mitochondrial quality, all kinds of things really lead to the upregulation of interferons. These can work on the same cell or in cells in the adjacent area, leading to a whole variety of signaling outcomes. And kind of straight down from the interferon receptor, you're going to activate things like STAT1, STAT2, as well as probably other stats to varying degrees. But in combination or at high concentrations, you can really get a whole variety of signaling complexes activated that then go on to have both pro-inflammatory and anti-inflammatory effects. So really, it is something where you could imagine that these are going to trigger the immune response. But anytime you unleash your army, if you will, you want to put in place some sort of mechanism to draw it back in. And this is a consistent mechanism within the immune system, and one that also is shared by these kind of inflammatory factors and type 1 interferons. And so just kind of how we got started and started thinking about interferons in the context of good or bad immune responses, if you will, was using the LCMV or lymphocytic choriomeningitis virus model of infection. This is a mouse model that one strain is able to induce an acute infection that induces a very strong pro-inflammatory environment, robust T cell activation, as well as activation across the immune system, B cells, innate immune cells. This leads to the induction of positive and negative factors that then ultimately lead to the control of this virus infection and immune memory. How is this dim or is that just me?
Doctor. See if just dimming the lights in the room will help.
Maybe by the end, it'll just go down and that'll be the end. Maybe. I don't know if it does. I don't even know how to fix that if it does. Oh, it's the brightest it goes. Well, we can all huddle around the computer here and just, it's very crisp here. Like the red is red. It's in some situations, as long as we could see it, in some situations you do get kind of this change from a pro-inflammatory, not totally gone, but to an anti-inflammatory environment. Many of these immune checkpoint and suppressive molecules that we've come to know that attenuates T cell function. These T cells are unable to finish the job of controlling the virus infection, ultimately leading to a chronic infection where you have these exhausted T cells. These are cells that don't proliferate very well, incapable of making many of the cytokines we associate with productive immune responses, have less cytolytic activity. These are then sustained throughout this chronic infection, and they still have effect. They're still fighting the virus. It's not like these are cells without function, right? Because if you deplete these, I mean, HIV and AIDS would be the best example. If you deplete these, virus loads go right back up, right? So they've reached a homeostasis where they're not able, or equilibrium, where they're not able to control the virus, but they're still maintaining some control. And so a lot of kind of therapies, the idea would be, how do we change this into this? And to do this, as I mentioned, we use two LCMV strains, clone 13, that generates a chronic infection, LCMV Armstrong, that generates an acute infection. These are very similar, 3 amino acid differences, no change in the epitopes, recognition. Really what it is this virus has a higher affinity for its receptor, alpha-dystroglycan, so it can get into cells better. And once in cells, it has a polymerase that replicates faster, so it can both get in better, replicate faster, outpace the immune response, leading to this path of virus infection. And so in initial experiments, well, for me, it seems like a very long time ago, what we observed was that, and maybe not unexpectedly, if you looked at a variety of type 1 or just interferon-stimulated genes, this is microRNA for maybe a couple people in the audience would remember what that is. What we essentially saw is that as you get high levels of ISGs, as the infection resolves with the acute, they go down. As the infection progresses into chronicity, they remain at elevated levels. Mind you, we can't measure interferon proteins like alphas or betas at this point. We know they're there because if you block things, things change. But this is really kind of an immeasurable amount of interferons that still have a pretty big effect. And what we observed was that if you use either a type 1 interferon receptor knockout mouse or a interferon receptor blocking antibody, that you could bring down levels of IL-10. IL-10 is an interferon-stimulated gene. You can bring down levels of PD-L1, also an interferon-stimulated gene, but you had higher virus titers. And so this all makes sense that these are interferon-stimulated genes. These go down. These go up because of less virus restriction. However, this for us was interesting because it was the first time that we had ever seen an uncoupling of virus titers and type 1 and these expression of these inhibitory receptors, suggesting to us that, and we went on to show in many ways that interferons are really serving as a kind of a communication between kind of the virus host to say how well the immune system is doing at fighting that infection and translate this into levels of these immunosuppressive molecules to attenuate the immune response. And there's many of reasons probably for that, but that's what's happening. And in fact, if we block type 1 interferons early in infection, so this is blocking kind of at the onset of what would be a chronic LCMV infection, and then look 30 days later, we found, in fact, that plasma viremia and liver titers were both down, indicating that, I think, kind of unexpectedly, that if you prevent this early onslaught, high-level interferon signaling in this chronic infection, you could actually have a better, and this is immune-mediated, better immune-mediated control of virus infection. And this is true also, although not as well, but if you block later in infection, you can also see a decrease in virus titers. And so what is the mechanism? And I think there are a lot of them. A whole lot of things change if you block interferons, type 1 interferons, early or late. But for the purpose of this talk, one of the mechanisms that we saw was an increase in interferon gamma. So this is the early blockade, an increase in interferon gamma if we block type 1 interferons. So, and we see this in a lot of our systems where there's this kind of antagonism, probably not direct, but antagonism between type 1 and type 2 interferon. And so a graduate student in my lab, Saran, wanted to ask, well, what are type 1 interferons doing in kind of the chronic phase of an LCMV infection? So we gave mice LCMV-specific transgenic CD8 T cells, P14 cells, infected and then blocked with either a type 1 or interferon gamma or both antibody in the midst of the chronic infection. And he focused almost singularly on virus-specific CD8 T cells. And so just by way of explanation, this new kind of complexity to these virus-specific CD8 T cells or exhausted CD8 T cells in chronic infection, also seen in cancer. You have these precursor exhausted cells. Everything here is exhausted, all activated, all exhausted. You have these precursor exhausted cells that can proliferate, self-renew, as well as spin off a little more effective exhausted cells, and then terminally exhausted cells, which are not without function, but again, these are the least functional of all these. And what he observed is when he blocked type 1 or type 2 interferon, he saw an increase in this population of self-renewing kind of these progenitor exhausted cells. This went up with IFNAR alone, but really went up when you block both IFNAR and interferon gamma together. No real effect with interferon gamma blockade alone. And this translates numerically. You could see that there aren't many of them to begin with, but you get this bump if you block type 1 that really is accentuated if you block both type 1 and type 2 interferons, kicks them into cycle, which I think is to be expected. But when we looked at the terminal exhausted cells, we saw, in fact, that they now had less function. They weren't numerically too different, but they were now much less functional. So you had a situation where type 1 and type 2 interferons were affecting both, so negatively affecting these precursor exhausted cells by preventing their proliferation, but sustaining these effector exhausted cells, right? So how is that happening? We're still working on a lot of this, but one of the things we wanted to ask is this a direct effect? So we took cells, so PD-1 positive CD8 cells at this point. So these would be virus-specific CD8 T cells by and large. We then sorted them out, deleted with CRISPR, either type 1 interferon, interferon gamma receptor, or both, and put them back into infection-matched controls. And so if we delete these out, you could see that IFNAR goes down, gamma receptor goes down, so that the CRISPR works. Good. We saw that if you transferred both of these, there was a decrease in the overall amount of cells in the recipient. So you need both of these. Again, that idea of you need these to sustain the function of these CD8 T cells. However, when we looked at their differentiation, we saw that really whether they lacked interferon receptor or both, there wasn't really a dramatic effect. I know this looks like it, but he won't give me anything else. But if you look at the percentages, it actually is the same across the boards for these. So you don't see any change in the TPEX. If you look at the numbers, again, nothing, maybe a little bit if you really wanted it to be, but really nothing. So no change in these. So it seems to be that blocking interferon and interferon gamma receptor or gamma are having an indirect on these CD8 T cells that allows these cells to proliferate. And so we're currently determining how this works. There's some suggestion that it has to do probably with changes in the macrophages, dendritic cells, and their antigen presenting capacity, as well as their localization with these subsets in the kind of follicular region, at least in the spleen. But we're trying to get at that now, how that's working. But I kind of wanted to show some other stuff, so I'm not going into this too much. But when we came here to Canada, we being, I guess, me and my lab, decided that we wanted to also start looking in the cancer setting and try and understand how interferons were affecting tumor growth, the immune response during tumors. And, it had been established, certainly before this, that interferons are important really at all stages of cancer development in different ways, certainly, but everything from the initial immune evasion and restricting that initial immune evasion to all the way around to ultimately metastasis and kind of priming these cells probably to go to the lymph node and allowing them to metastasize after that. So they're important for both I think this is out of order. Yeah. Interferons are critical for the beneficial response to cancer therapies, radiation therapy, immune therapy. You need these interferons, but through the upregulation of a variety of suppressive factors, and other mechanisms, they also can drive the resistance to these therapies. So much like I was saying before, you initially need them, but then they ultimately restrict the ability of these therapies to keep working. And two recent publications, not by our group, came out that showed that if you incorporate a JAK inhibitor, so something that's going to block type 1 and type 2, as well as other things, but it was pinned towards type 1 and type 2 interferon. If you incorporate this with anti-PD-1 blockade, that you can have a much better responsiveness during to lung cancer as well as Hodgkin's lymphoma. So by lowering interferons, probably other things, but by lowering interferons, you could now get a better response to anti-PD-1. And this was kind of consistent with what we had seen in these virus models. So we wanted to ask what determines these opposing outcomes of type 1 interferon signaling, and can they be uncoupled? And one of the things that we started to look at was a transcription factor called IRF2. IRF2 is interferon regulatory factor 2. It's constitutively expressed in cells, pretty much all cells, but is upregulated by interferon. So it itself is also an ISG. It's most notably associated with preventing the function of IRF1, IRF1 being a main effector of interferon gamma and type 1 interferon signaling, but also can affect other transcription factors, and generally is thought of to work in a negative fashion, to suppress kind of interferon-induced responses. But like anything, it can also have a positive effect. And it had been studied pretty much pretty well, I think, in cancer cells. Again, like everything we're seeing with cancer, higher IRF2 expression can be associated with better cancer growth in some types of tumors, worse cancer or better control in other types of tumors. And mechanistically, how this is happening comes down to, I think, other factors within those tumors. But really very little was known about how IRF2 regulates the immune response. So a postdoc in my lab at the time looked into this. We got IRF2 sufficient mice, or rather IRF2 knockout mice, and we gave them IRF2 sufficient tumors. So it's not going to be a tumor-driven effect, whatever we see. We then looked for cancer growth over time. And what he observed is if you look at a pretty immunogenic MC38 adenocarcinoma tumor, you could see that in the beginning, the tumors grow about the same. So it's probably not, so it's not, we know it's not rejection due to having IRF2. However, if in the wild type mice, whereas they continue to progress, the IRF2 deficient mice now are able to control these tumors, with in fact many of these tumors looking like they're gone, right? Looks like these would be complete responses from a kind of measurement standpoint. And we see this in a very aggressive kind of non-immunogenic B16 melanoma, as well as a breast cancer model, that if the mice lacked IRF2, they were better able to control these tumors. So what cells mediate this control? And we tried a variety of different cell types. We created a IRF2 flox mouse. And the one that worked is if we floxed it out of only CD8 T cells. So only CD8 T cells in this mice lack IRF2. What we observed is that now they were able to control the tumor much better than the wild type or littermate controls. So it turns out it's not as good as like a complete IRF2 knockout, suggesting that it's not just CD8 T cells, but predominantly CD8 T cells are mediating this effect. And in fact, this is continual, because if you wait until the wild type mice would have kind of had complete or total, kind of they had to be euthanized, and then deplete CD8 T cells, you see the tumors pop right back up. So this is not an elimination of the tumor, but a ongoing control of the tumor by these CD8 T cells if they lack IRF2. So how is IRF2 suppressing CD8 T cell function in the tumor? So there's a variety of ways that it's probably doing this, one of which is when we look at IRF2 deficient CD8 T cells in the tumor. This is an IRF2 knockout, but we see the same thing when only CD8 T cells lack IRF2. You see that whereas the wild type mouse has this really bright double positive 39 lag3 PD1, so double positive or triple positive for all these kind of negative immune regulators. In the IRF2 deficient mice, they kind of sit in this CD39 positive LAG3 dim PD1 dimmer subset, which has been associated with better effector function, so sustained effector function as opposed to exhaustion. And if we in fact look at the tumor-specific responses of these CD8 T cells, you see that in the IRF2 deficient mice, they have higher levels of interferon gamma, somewhat of TNF alpha, but it's really coming from this kind of mid population here. So these cells are able to go into the tumor, and if they lack IRF2, they're able to maintain their function. They also have higher levels of a variety of other things that you might associate with functional T cell responses. I think not unexpectedly, certainly they had higher levels of interferon signaling. So if you look at RNA sequencing of the IRF2 versus wild type, so up means it would be up in the IRF2 deficient CD8 T cells, you see that they have much higher levels of interferon signaling, type 1 and type 2, which would be consistent with not having something that dims interferon signaling. However, again, this is usually what we would see in very exhausted CD8 T cells. However, here they're not as exhausted, and they actually function to rely on interferon. If you deplete either separately or shown here, both block interferon type 1 and type 2 at the same time, you see, again, you very rapidly lose this control of tumor. So it's something where these CD8 T cells are going into the tumor, they're resisting an exhaustion, and then able to interpret this interferon signaling now, type 1 and type 2, into positive responses as opposed to the suppressive ones. And so what we wanted to do is now, and we're moving forward with this in this kind of a setting, is we wanted to ask, can we use this system for adoptive cell therapy? So we did this first in the mice. We have tumor-specific CD8 T cells here. Okay, out of order. So we have tumors that have a CD8 T cell antigen to which we have a transgenic T cell, put those into mice. We can then take these tumor-specific T cells, delete out IRF2 or not, and then activate them briefly and then put them back into these mice. And what we see is that, again, you get a not very much nicer tumor control with very few. This turns out to be very few. In fact, you need 2 to 3 times more of these to even have an effect. So with very few of these cells, we can now see a much better tumor control. And in fact, we're now adapting this to human, and we can delete this out, and we're putting these into kind of humanized models of CAR T cell therapy with the hope of being able to maybe move this into the clinic if it shows some promise. And so essentially, just to kind of go through it, because I just did, interferons increase IRF2, leads to activation of the cell, but then diverts kind of away from this functional response to an exhausted response that's unable to clear the tumor. If you delete IRF2 in these CD8 T cells, you can now get sustained effector function that gives better longer tumor control, better response for adoptive cell therapy, and it turns out better response for anti-PD1 as well. And I think what's interesting here is when we looked at these IRF2 deficient CD8 T cells, they had the same level of T cell activation, TCR activation, as is observed in the wild type cells. And TCR, kind of levels of TCR activation are certainly important in the induction of exhaustion, but in many ways are thought to be one of the main, like the only factors in the induction of exhaustion. And I think this suggests that there are kind of multiple ways. One certainly coming down through the kind of direct antigen sensing or direct antigen stimulation, but also a mechanism whereby the cells are kind of interpreting the environment and understanding levels of inflammation and then modulating their responses as such. So we're now trying to figure out what some of these relationships are and pathways of exhaustion and how they either converge or diverge to lead to kind of the inability to control tumors. And so moving on a little bit, we next wanted to ask, well, how is interferon signaling related to PD-1 therapy immune outcomes, and can it be used as a biomarker? And so these are the wonderful people that did all of this work. I just kind of stood there and I didn't do anything. But I'll pretend that I did, and I'll present it if I did. But they really did all the work. So Giselle really came in with an idea. She was interested in how interferons could be used to, or how cells respond to interferon as a predictor of immunotherapy outcome. And that's not a novel idea. Many people have tried it. It doesn't seem to work. Sometimes it does, sometimes it doesn't. But her thought was that perhaps it had to do with the idea that, the bulk approaches aren't really the way to go, but instead it would be the complexity at the single cell level, or at least the single subset level, that would really kind of identify something about these immune populations and how they might be able to respond to therapy. And so, really, the idea would be that type 1 interferons can act in one cell to give a negative response, another cell to give no response, another cell to give a positive response. If you were to bulk this all together, it would just be a net zero, right? However, if you were to look at these individually, you might be able to determine something about the biology. And so she didn't set out to do it this way, but it turned out because of COVID that we did it this way. She was able to get patient PBMC. So these would be patients who would ultimately go on to have anti-PD-1 therapy. They're viably frozen, thaw them, stimulate them with interferon beta or not, and then run CyTOF and look at various subsets of immune cells. And, it's not hard to believe. You sprinkle interferon beta on cells, they all pretty much react. These are different interferon stimulated proteins that she was able to stain for. So this is all human stuff. I forgot to mention that. But different cell types seem to upregulate different of these interferon stimulated proteins to different degrees, right? So she generated a interferon response capacity score, which took this into account. It's essentially a fold change of stimulated versus unstimulated in any given cell type, taking not a weighted average, but taking into account the overall pattern of these upregulation of interferon stimulated proteins. And in most cell types, again, everything goes up, but there was no clear kind of divergence in anything. However, when she looked at CD4 and CD8 T effector cells, what she observed was that they really stratified into patients that responded robustly to type 1 interferons. So again, kind of a fold change versus those that responded less well. Again, everybody responds. It's whether you respond strongly or weakly to this. And when she looked prior to therapy, so before in samples from pre-therapy, and asked, well, how does this high or low interferon stimulation in the patient's cells reflect outcome? What she observed was that in the first cohort, this consists of head and neck and melanoma samples, patients whose CD4 or CD8 T cells responded less strongly actually had better overall survival than patients who responded more strongly. And this was true in a melanoma validation cohort as well as in a lung cancer cohort. So this was kind of striking to us. I mean, not really if one takes into account kind of what we had done in the virus infection, but really when, the field is that in the cancer, a higher inflammatory environment is associated with better outcome to anti-PD-1. But again, those biopsies or those samples are going to be bulk where they have tumor cells, probably predominantly tumor cells, and then other cells in there, right? So if you look in the immune system, it appeared to be, at least in the periphery, and in terms of how they would respond, that less overall interferon responsiveness was a better hallmark of outcome for anti-PD-1 therapy. And we went on to show that mechanistically, this is not due to either of these groups having baseline differences in the level of interferon stimulated genes. So we did RNA sequencing. So in fact, there really aren't many interferon-stimulated genes being made. I mean, this is the periphery. You could imagine it's kind of like a highway, right? So they're not really interacting with anything. They're just kind of shooting through. You don't see a bunch of interferon-stimulated genes. However, when she looked at the epigenetic profile, so did ATAC-seq and looked epigenetically, what she found is that the patients who had high responsiveness to interferon were epigenetically poised. They had their open chromatin regions were enriched in interferon-induced transcription factors, STAT1, STAT2, IRF1, IRF2. So they were poised essentially to respond to interferons and do so robustly. Why that is, I don't know. These are hard things to, in my mind, almost impossible to figure out in human samples, and we don't have a good mouse model for this. Had a lot of ideas about this, but actually how that is happening, we're not sure. But it was clear that there was a difference and that these cells that responded strongly were ready to do so. And so we asked, well, okay, so what's happening in the tumor? And so we got a hold and reanalyzed some published single-cell RNA sequencing data sets. And when you look only at immune cells, so again, no tumor in this sample, and that's, I think, I don't know, but I think key. If you look at only the immune cells, you see again that this is colored by the upper kind of third, the upper level of cells that are expressing interferon-stimulated genes. And if you, and this is both type 1 and type 2 interferon-stimulated genes put together, if you look, you see that responders have a much higher level of cells that are expressing high levels of ISGs or non-responders than you do the responders. So similar to the responsiveness in the periphery, in the tumor, the patients who were going to be responding seem to have baseline levels that are lower of these ISGs. And this is not like being driven necessarily by one cell type, but if you look across cell types, you can see that generally the responders in blue, most of the subsets have lower levels of interferon-stimulated genes if they're in that responder phenotype or group. And also, if you take all this data and just collapse it down as if it was bulk RNA sequencing, what you see is that if you look for upstream regulators, so this is in Ingenuity, it's just kind of like a pathway analysis, except it boils it down to a molecule instead of a pathway. It's not really that, but that's kind of fundamentally how it's easier to think of. What you see is that in the non-responders, the main upstream regulators are, in fact, interferon-driven genes, interferons or interferon-stimulated or stimulating genes, whereas in the responders, some of the most upregulated are actually genes associated with attenuating interferon responses in the kind of protein translation, RNA regulation. Certainly TREX1 that would be involved in DNA degradation to prevent STING activation. So you get kind of this dichotomy where the non-responders are really enriched again in these interferon-stimulated pathways. And so if we go and look, and kind of one of the things that stood out to us was how interferons were spread, signaling was spread across kind of all these subsets. So we devised an interferon 1 and type 1 or type 2, so gamma signaling score. And these were really made in a variety of ways, largely in the interferon database, made up of genes that were, for example, stimulated, I don't know the exact number, but stimulated 5-fold by type 1, but only 2-fold by interferon gamma and vice versa, because there's no actual clear cut. And so we formed these, and what we saw is that, again, you saw that the upper third of cells with this interferon, type 1 interferon signature fell across all the different immune subsets, but not in all. So it wasn't that every CD8 T cell had this. It was across that CD8 T cell subset. Same for type 2. And if we look, there was about a 60% overlap, give or take, between these two signaling cells, between the signaling of the, yeah. So when we look kind of to further kind of put together this idea that it's not just, naive T cells are receiving interferon signals. When we look at the CD8 T cells, recluster these into different differentiation subsets, what we observe is that, again, there's this spread where in about 40% or 33% of the kind of all the different CD8 T cells are in this highest level of interferon signaling. Right. So it's not one subset, it's not one within one subset, it's not one differentiation state. And so what this suggested to us is perhaps instead of interferons being kind of just in the tumor kind of everywhere and everybody interacting or made only by a certain cell and you happen to be the one cell that can interact with that cell, that maybe there was some sort of spatially distinct area where interferon signaling was occurring. And so what we did is we got a hold of some publicly available data sets for Visium. So this is single cell, not single cell, this is spatial transcriptomics. The resolution's not great, but good enough, I guess, for preliminary experiments or to get us kicked off in this direction. And what he observed is if he looked at that at an interferon score, so again, roughly based on the interferon score I had just been showing you, and looked in human skin cancer, lung cancer, or breast cancer, he saw, in fact, that there were these distinct islands or patches of where interferon signaling was occurring that dissipated out to areas where there was not kind of robust levels of interferon signaling. And in fact, and I think to my surprise, when he looked at various mouse models, he saw the same idea. I thought B16 would just be a blob of tumor, big black tumor. But it turns out that there are, in fact, also these kind of regions. And if you look by immunofluorescence, it really stands out that you get these really defined regions of interferon signaling. And again, there's overlap, not complete, but overlap with type 1 and type 2 interferon. And if you look again at a different Visium sample, if you look at the areas of this interferon signaling and overlay it kind of with areas of where the tumor is, what you can really start to see is that this idea that really where these interferon signals are, so the interferon signals are the things that are jutting up in the air, and over colored is where the tumor is, you could see that they're really at these kind of border sites often of where the tumor is where you start to see these interferon kind of regions or patches. And so he did a immunofluorescence stain. This is an interferon stimulated gene 15. This is a kind of a canonical ISG. It's robustly activated by type 1 interferons, much less so by interferon gamma. And what he observed is if you look in a whole kind of tumor section, so this is a melanoma section, you could see again, there are these kind of splotches where interferon signaling is. And in some tumors, there's more, and some tumors are bigger, and some tumors are smaller, some tumors are less. But you can always see this kind of splotches of it. It doesn't seem to be just a blanket of interferon signaling. And if you focus in on two of these, what you could see is, so this is now imaging mass cytometry of that same tissue. And what you see is that you can't see. Blue is the MART1. So blue is the tumor cells. And you can see now that there are these regions where there's interferon signaling. This we're not sure. We think this may be a newly forming or a resolving interferon area. We're not sure kind of the dynamics and probably won't be able to get that with static imaging, hopefully maybe with the mouse. But if we focus in on one of these sections, you could see that, two of these ISGs are predominantly overlap in their expression. However, if you look at another one, IDO, or I think PDL1 has a similar kind of idea, you see that now, yes, it can be in where these interferon other stimulated genes are, but it's not in all of them, and certainly in other places. So there can be differences in the expression. I like to think it will be the positive and the negative, but I suspect it won't be quite that clear. But certainly differences in how some of these ISGs are expressed in this tumor microenvironment, and we can localize it to other cells. And in fact, we're now starting to be able to tease apart these regions and then ask, what is the cellular composition in this region? And I don't really have more data on this. I'm just so excited to show it that I figured I would kind of show it and give an idea of the directions we're going into. We now have, I think, 10 or 12 melanoma samples, and then we have as well as an additional 3 responders, 3 non-responders before and after anti-PD-1 therapy, where we can do this same type of stuff as well as some of the spatial transcriptomics we've done on this and are muddling through trying to get that analysis going. So I think it'll be really interesting to ultimately figure out what is happening in these regions. Are they, for example, if I say that there's less interferon signaling in a patient who responds to immunotherapy at baseline, does that mean that there's less of these in those patients? Does that mean that they're smaller than they would be otherwise? Is there a difference in the cellular composition in these regions? If those papers turn out to be, I think they'll be correct, that the JAK inhibitors can actually have an effect, this is presumably where they'd be having their effect. So what would it be that they are dimming down? And we can now hopefully start to use some of these mouse models as well to tease apart how these regions are being regulated. Are they driven by type 1, type 2 interferon? Kind of all the questions you might imagine we would ask. We hope, I don't, you're probably all smarter than me. So hopefully we'll ask all those questions and be able to really get at what's happening in these areas. And so kind of in conclusion, showed that altering IRF2 expression can really change the induction of the exhaustion program. So really kind of filling this switch of something that is going to turn from productive interferon responses to what we would call negative interferon responses. And we're currently trying to see if this is something that could be viable for the clinic. In the periphery and in the tumor, in immune cells, decreased interferon responsiveness and signatures are associated with better anti-PD-1 efficacy, and which is consistent with this trial, right? Suggesting that patients with lower, artificially or naturally lower levels of interferon signaling are better off when it comes to anti-PD-1 therapy, and that these interferon regions are spatially confined within the tumor, and they really form kind of their own ecosystem, we think. We think there'll be different cells there than there would be necessarily in other spots. It's conjecture, but that's kind of the hypothesis we're going with. And we think they may be the battle sites really where the immune system is encountering and ultimately fighting the tumor, and where those kind of decisions are being made, right? Upregulating these negative regulatory factors, downregulating the immune response, ultimately allowing for tumor growth. And so conceptually, I think there's this dichotomy, right? Where you have in just normal tumor progression, I think high inflammation, high interferon signaling is a good thing. Think STING agonists, right? You haven't translated well into the clinic, but certainly in mouse, by amping up the amount of inflammation, you actually can get a better control of the tumor. However, in immunotherapy, it seems that that's a negative. And now you want to either artificially, so therapeutically, or patients who just naturally have lower interferon signaling will be better off for anti-PD-1 and potentially other therapies. And we're looking into some of this now. So really you get this dichotomy wherein, it's not that you want high or low, it's that you want high or low depending on what you are doing or what the hope is, what the basis of the tumor is at that time. And we're now trying to understand really the conceptual difference and why both of these would be good, but good in different situations. And so with that, I'd like to thank both current and past members. Maddie's here. I don't know if she's in the audience. I can't. Oh, okay. Well, Maddie's here. And actually, I think you probably did some of the work I showed up here, as well as Matt used to be in my lab, but he quit. And then other people who have really helped with this, both Sabello and Giselle, and then Wen, who's taking over a lot of this other stuff, as well as all our collaborators and funding. So thank you very much.
I'm hoping you're willing to entertain a few questions. So questions, please. Marco, why don't you start?
Thank you, Dr. Brooks. That was really interesting. I had one question about the epigenetic timing data you showed. Was that observed across all PBMCs? Was that in T cells specifically?
So it was four responders, four non-responders. So it was not a huge sample size, but was consistent. It was observed in both effector CD8 and effector CD4 T cells. We didn't really analyze the other too much because that's what we needed, and then she got a job. So that's kind of how that went. But yes, in effector CD4 and CD8 T cells is true in both, which I think is interesting because they're also very different cells in many ways.
Thanks.
I think Eva had her hand up first, but I got you. Go ahead.
Fantastic talk. Thank you so much. This was the most enjoyable T-cell talk ever heard. I have a couple of questions actually. So you're talking about type 1 interferons, and did you ever really look at the difference between type 1 interferon like alpha or beta, and if that relationship changes as you see this flip in the immune response?
We haven't, because you can't really detect them very well in our assays. In the mouse, we just now, Ali Abdul Sader, who's at York, made an interferon alpha knockout. So he knocked out the whole locus. So we now have the ability to ask those exact questions. We send the mouse for what's the contribution of alpha, what's the contribution of beta, and then also start to parse into different cell subsets. What's the contribution from this cell type or that cell type? Because at least in the xenium data, which I didn't show, we can see beta. It's probably an underrepresentation, but we can see it. And we see it in like CD8 cells. And I don't really understand that, but it's there. So we can start to test questions about how that might be, would their contribution, because I love the idea that they're the ones who would be going into these areas, making it and kind of stymieing their own responses by upregulating these checkpoint molecules.
Yeah. And then is this exhaustion that you're seeing, is that limited to antigen-specific T cells, or is it all T cells, or is it maybe?
So it depends. So in these cases, it's generally not a systemic, like a system-wide exhaustion. It's an antigen-specific exhaustion. However, when you try to mount a secondary response, so we did this with Maz. I think he was on that paper. Wasn't he your postdoctoral mentor? No, maybe not. Oh, I guess not. So we could see that the secondary response to a pathogen, if you have this chronic infection, is very different. So it's not an exhaustion per se. It's a difference in the differentiation of those responses that leads to different outcomes. TB is worse. If you take a chronic infection in the middle of it, give it these cancer cells, the outcomes were you get a very different CD8 differentiation, and they can't control the cancer very well.
Yeah.
So the exhaustion, it's so a long-winded answer to maybe a short question. The exhaustion seems to be like tumor or virus specific, but it has effects on subsequent responses, secondary responses.
No. Go ahead.
The spatial location of those interferon producing cells in the tumor, the last data that you showed, they're pretty cool. And so it's like, do you have any indication that these cells are associated with metastasis?
So no, because we don't have the ability to really to know that now. So these are everything we're working with really now is stage 3, usually stage 4 cancer. So they probably all have Mets. So whether they're associated with or not, we can't tell. And the mouse models won't tell us that either.
Yeah.
So I don't know. It would be great to be able to kind of piece this together kind of sequentially, something like a very early kind of identified tumor, which you can do with melanoma. And people are doing that in other kind of areas. But so I don't know, but it would be cool to know. Yeah.
David, go ahead. Yeah, thank you for your presentation. It was really interesting. I have a question about the interferon pathways in CAR equipped immune cells. Do you anticipate that the immune profiles are going to be different? The interferon profiles will be different with CAR T or CAR NK and various other forms of immune cells?
In terms of
For their ability to change some of the landscapes you showed us so elegantly at the end of your presentation.
Yes, I think they'll be different. There's some publications out there suggesting that, kind of exhaustion in those cells is also associated with kind of higher interferon signaling, whether that's causal or just an association, I don't think is so clear. They'll go in a very different state, though. So I would imagine it would be different, right? They're in vitro activated and expanded and all sorts of things. So I think they would go in. How they respond to interferon, I think would ultimately be the same, that with time you would get this interferon-induced suppression. And that's the idea behind kind of deleting IRF2 in those cells to let them. That said, there's a clinical trial going on here where the CAR T is actually a JAK-STAT kind of thing. And I don't know the outcome of the clinical trial outcome's not clear, but certainly in preclinical models, that makes for a much better CAR T cell. So that would have heightened interferon signaling. So the answer would be, I'm not sure, but I think in just a regular CAR T cell that you would see this interferon kind of over induction exhaustion type of phenotype.
My question today is in your IRF2 knockout experiments, you knocked it out in monoclonal populations, I guess, of T cells, like antigen specific.
Yeah.
Yeah. So that's why you can see it applied to CAR T therapy. But for a better translational value, can you envision doing it in polyclonal anti-tumor population?
Well, that's what we're doing with the human. We'll get PBMC, sort out the T cells. We have a CAR construct we can load into them and knock out IRF2 in this way. And then that would be the goal to test that. Certainly we know in vitro that they can perform fine, but in vivo, kind of for what these humanized mouse models are worth, I think that that's where we'll test that.
T cells, like CD8 positive T cells, can you get and knock it out in those?
Probably. We have certainly in broad populations of T cells, we can knock out IRF2. We just haven't we've kind of moved on from a lot of the mouse studies when it comes to that and put it more towards the human idea, because that would be our interest for that kind of branch of the studies. In the mouse, we're kind of more focusing on the biology of how IRF2 is affecting these responses. But I know we can, in a kind of a polyclonal CD8s from a naive mouse, we can knock out IRF2, stimulate those cells, and they'll proliferate. We haven't put a CAR receptor or done any of those kind of studies that you're asking about. You probably could. I mean, the precursor frequency of these, CD8s is going to be somewhere in the order of 50 to 100, maybe 150 cells. They'll amplify a lot, but we've done those kind of experiments where we take naive cells, put them in, and you do get a response. But I think, we haven't done that. I think if we did it, we would use a CAR construct or something that would be using. That said, I think for some of these TCR engineering and all these kind of things, I don't see why it would necessarily, I think it would also work in that setting. If you had a TCR specific to, MART1 or specific to some of the HIV or, whatever kind of one's interest is, I think it would also work on that setting, certainly because these P14s are using a TCR and not a CAR construct. But I think in a way to maximize the amount of antigen-specific cells for an experimental system, it's a lot easier to have a lot of them, essentially.
Yeah, that was great. Very refreshing, and we used to work on interferons, so it's kind of back in time. So are these cells also called memory inflationary T cells? Is this the same population? LCMV induced?
No, it's not. So it's not CMV. It's unfortunately CMV right there in the middle, but it's not like a herpes virus. They are cells that, so when you get these TPAC cells, these progenitor exhausted cells, when you get these virus specific, or it turns out cancer specific CD8 T cells activated, they differentiate how they get to these. I'm not sure is exactly known and certainly debated, but you get kind of a couple populations, one of which is this TCF7 or TCF1 high CD8 T cell that's exhausted. They're activated. They're not inflationary because it doesn't take all this time. They're there within the first few days of activation. And they have an ability to self-renew, whereas the more terminally exhausted cells don't really. And these are the ones that, for example, when one gives the anti-PD-1, these are the cells that are thought to be targeted and then spin off progeny that go on to fight the virus or the cancer. So they're not like an inflationary memory that kind of comes with long-term kind of antigen stimulation. There's something that's formed almost immediately. And if it were an acute infection, this phenotype of cells would be what would go on to form memory.
Constitutive interferon activation that we see in cells with DNA damage, repair gene deficiency such as BRCA or or any in those pathways, and other cancer cell associated mitochondrial DNA and STING activation or cytosolic nucleic acid sensing. So how do those fit in your human scenarios when cancer cells may have such mutations, and do you see the constitutive ISG between them?
We do. We are only getting snapshots of this, but we definitely in the tumor cells, absolutely. What's interesting is in the tumor cells, it's not everywhere, right? Like if you saw that big picture, that was a big tumor, like that's all tumor, right? And it's there was still just spots. So a lot of those spots are melanoma, but a lot of the non-melanoma cells or of the melanoma cells are not expressing these. So I don't know. The one kind of hint we got from two, so again, with a grain of salt, from two of these kind of slides are this huge SOX1 upregulation, which is a negative regulator of interferon signaling in the melanoma cells that were not expressing high levels of these kind of ISGs or ISG15 at least. So that's kind of the thought that maybe that's inhibiting it, but again, that's a small sample size, and we should know more soon. So I don't know. I think every cell type, like why would a CD8 be making interferon, type 1 interferon? Like you go back in publications and apparently they haven't done it since like 88, right? So it could be. I mean, it could be in there, but endogenous retroviruses are always kind of the culprit that's brought up for how that may be acting. Could be some sort of transfer. I mean, there's any number of ways, but why? So we'll see. I think is at least our goal.
Great talk. I just wondered with your plans for the CAR with the IRF2 knockout, are you thinking about doing that in the context of solid tumors where they haven't been terribly effective? Like, do you think that could make some of the difference between persistence and just exhaustion as soon as they get there?
That's exactly what I think.
Yeah, okay.
It'll vet out one way or the other, but that's exactly what I think.
Okay.
I think that a liquid tumor would be less likely just because,
Yes.
yeah, because it doesn't have that kind of dense, I mean, it does, but that same kind of idea. That's at least our thought.
And then are you planning to add some of the other edits that people are doing to make them like switch receptors and some other things?
If we get there,
okay.
I mean, it really all depends on kind of how these models turn out. And if they turn out well, then I think, yeah, especially with, people around Princess Margaret who are doing all these kind of things, I think could be awesome. But right now, I just want to see if
Yeah. You got to start somewhere.
Yeah.
Awesome. Thanks a lot.
Thank you.
I think that should do it for tonight. So join me in thanking Dr. Brooks for an amazing.
Thank you.